Today, I'm happy to welcome Dr. Ian Bird to Grand Rounds. Dr. Bird is a professor here in the Department of Obstetrics and Gynecology, and he also holds an affiliate appointment in pediatrics. With a PhD in biochemistry, Dr. Bird came to UW in 1994 to the Department of OBGYN, where much of his research has focused on vascular endothelial function in pregnancy, the placenta, and preeclampsia. In addition to a long history of grant funding, publications, and recognition for this work, Dr. Bird has served as the Vice Chair of Integrated Graduate Training, the Director of Endocrinology and Reproductive Physiology Graduate School Program, and the President-elect of the Perinatal Research Society. He has received honors in this role, mentoring numerous scientists as they have established their research careers. And so please join me in welcoming Dr. Bird this morning.
All right, thank you. Hopefully people on Zoom can hear me as well. I've got no ability to see that. I'll start by saying that what this actually is a presentation we gave to NIH as a proposal for a program project grant. So it is by definition going to be technical. But don't worry if all of the terminology or the details are not fully things that you're familiar with. We've tried to weave into it the overall point of what we propose as a mechanism for preeclampsia. And to those of you who are clinicians, I would say that, which is obviously the majority of you, I would say that think of it this way. This is a proposal that unifies the inflammatory origins of preeclampsia with the endocrine and vascular origins of preeclampsia. That's what's novel about this work. So we're putting the two together. It's not the immunologist versus everyone else. This is how everything comes together in one coherent story. Okay. So hypertension and pro-hypertensive diseases in general, even beyond pregnancy, are all Th17 associated conditions, T helper cell 17 conditions that are associated with also an elevation of Th1 immune cells dominating over Th2 cells. So the Th2 are pro-inflammatory, sorry, the Th1 are pro-inflammatory, the Th2 are anti. Th17 is pro, Th10 is anti. All of these diseases, and that would be hypertension, obesity, rheumatoid arthritis, Crohn's disease, etc. All of these diseases all begin with an enhanced interaction between a T cell and a target cell that is something else. That could be a bone cell in the case of rheumatoid arthritis. It could be endothelial cell in hypertension. These Th17 associated diseases are also, like preeclampsia, associated with cytokines TNF, interleukin 1b, interleukin 6, and interleukin 8. They're all elevated in pregnancy anyway, but they're more elevated in preeclamptic subjects. Preeclampsia is characterized by early endothelial dysfunction and failure. Even before any clinical presentation, as early as 8 weeks, you can actually show that vascular function, endothelial function is impaired. The enhancement that normally is seen with pregnancy is just missing. So others have recently shown that exposure of endothelium to these same cytokines, TNF, interleukin 1b, 6, and 8, can actually reproduce the endothelial dysfunction of preeclampsia. So these clearly are mediators or potential mediators of that failure. When you bear in mind that pregnancy is a 9-month condition, you essentially have a negative input to the endothelium long-term. And it's no surprise that while in the first few weeks of pregnancy, everything is going along just fine, literally 1, 2, 3, 4, 5 weeks, everything is fine. And then progressively it goes downhill until it clinically presents in the middle of pregnancy. So this leads to our central hypothesis for this proposal, which is that the adverse interaction between immune cells and endothelial cells in hypertensive episodes of pregnancy results from prior changes in cell states, T cells, but also endothelial cells, to enhance the negative regulation of endothelial function and to drive the immune cell population towards Th1 and Th17 populations. We think both of these things are key, and that you get into a feed-forward loop where the more the endothelium becomes dysfunctional, if it had a way to exacerbate the T cell state towards Th1 and Th17, you would end up in a runaway loop that over time would get out of control. And then in the clinical presentation is where it's cascading into a bottomless pit of nothingness. I can't advance the slides. There we go. Okay. All right. So, significance of this from an NIH point of view is that causal studies, not correlative studies like how does this hormone change over pregnancy, but causal studies in humans are very difficult. In non-pregnant conditions in particular, because you don't know when the disease is going to begin. If you just try and study this in hypertensive males at the age of 50, you may have to wait 5 years to see the symptoms begin. Okay. Pregnancy, on the other hand, is different. It's a highly predictable state. We know within a matter of weeks when we're going to see these symptoms, if we're going to see them at all. So studying this condition in pregnancy not only has relevance to preeclampsia, it has relevance to generalized hypertension, and it has advantages. The availability of the human tissue in the placenta, in the pregnant state, the placenta, the decidua, and the cord, they're all disposable tissues, so there's no problems with IRBs. It doesn't impact clinical care in the slightest whether you analyze those. You have more problems getting consent than you do obtaining the tissue. And the availability of that tissue, along with the regular pre-delivery or preterm visits, offers longitudinal sampling opportunities. You can pick times through pregnancy to take blood samples and see how things are changing and compare the preeclamptics to the controls. And we have multiple resources in this department that are applicable. The first is obvious. We have a patient population. We have an ability to collect tissues and samples. But the other is that we have worked out and prepared and stored actually uterine artery endothelial cells from sheep. We have them frozen and we can use them anytime. So we can do uterine artery studies on an animal cell model anytime. Try to reproduce this dysfunction that's seen in humans. Likewise, we have HUVEC, isolated from human cords from both preeclamptic and non-preeclamptic patients, and we can compare those. And we have technological abilities and analytical machines in order to collect, isolate, and purify immune cells and analyze them by multi-dimensional FACS. And for those of you who haven't done FACS in a long time, probably when you started medical school, you could do maybe 5 antibodies at a time. We can do 28 now. That's pretty sophisticated stuff. So the program project has 4 projects, which I'm going to go through, and they will directly study a single cohort of pregnant patients. So one big advantage right off the bat is all 4 projects are done on the same subject. The data from all 4 projects can be related directly to each other. We can identify altered endothelial and immune cell states from normal, non-pathologic to pathologic states. And we can look at their changes as they relate to the preeclamptic or control population. We can relate that data to altered endothelial cell immune cell interactions. We can look at it in vivo, we can look at it in vitro. And we can look at multiple tissue levels. And if you look at the red pointer, the figure on the top right shows you project 1 is actually looking at endothelium in the uterine artery and in the umbilical cord. Projects 1 and 4 look at the cord, endothelium in the cord. Project 2 looks at immune cell populations in the decidua. And project 3 looks at immune cell populations in the systemic circulation with the obvious question, are they one and the same, or are they distinctly separate populations doing different things? Are the changes unique to the site of implantation, or are they actually circulating around the mom, propagating those problems into non-reproductive tissues? And then the third point is to establish longitudinal changes in Th17 population in hypertensive versus preeclamptic pregnancy to see if any of the circulating either cell or endocrine markers could be used as a better predictor of an adverse clinical outcome. We already know that there are plenty of endocrine tests, and we already know that they have very limited success. But what if you looked at the immune cells, which you have more success? So there is obviously synergy. I'm going to be brief on this. There's synergy between all of the projects. And they all interact in one other additional important way. They all work through what we call core C, which is a computational core. Much of this work uses multi-omic technology. We even get down to single-cell sequencing. And in fact, all of us get down to single-cell sequencing. And that requires very sophisticated data analysis. And this is the kind of data analysis, you can throw SAS away. I really don't care about a two-tailed t-test or an ANOVA anymore. It doesn't work. This is actually artificial intelligence analysis. This data is going to be analyzed by artificial intelligence. And the leader of that effort is Irene Ong, who we're very fortunate to have in the department. And she is the leader of Core C, which is computational core. Core B is the patient consent and tissue collections aspect, okay? So all of the projects work together and are held together through Core B and Core C. So the way we see the timeline of pregnancy is prior to 24 weeks. This is what we call the prediction phase. This is the period at which the first clinic visits and the follow-up visits begin. And then from 24 weeks, the outcomes phase. So the goal of the predictive part of this PO1 would be to be able to do a diagnostic test for biomarkers in the predictive phase to give you an accurate prediction of outcomes in the outcome phase. Okay, so you wouldn't just be reliant on ultrasound blood flow anymore or soluble FLIT, PLGF ratios. You'd actually have much more concrete evidence to say, I better see this woman more often because she's looking like she's going to go early preeclampsia. I give it about 4 weeks. She's either going to do it or she's not. So now's the time to step it up, rather than having her come in fainting, seeing spots and in a bad way, where it's already close to too late. All right, so project one is led by me. It's the uterine artery endothelial study. This picture at the top on the right, what it shows is that endothelial cells, these green bars are connexin 43 gap junctions between cells. And in a perfectly healthy, normal age, young woman who is able to have children but just hasn't chosen to do so yet, she has connectivity between her endothelial cells to a certain degree. But in pregnancy, that connectivity of the cells by gap junctions goes up dramatically. So we move from a non-pregnant state to a pregnant state, and you see a dramatic increase in the amount of connectivity. The gene expression isn't any different, but the connexins go up to the cell surface and become coupled. And this allows endothelium in a pregnant state. The enhanced vasodilation is because they can actually not only stimulate signals to drive vasodilation, but they coordinate them. Literally, if you look at the calcium responses in these cells, if you look at a field of cells and they flash on and off, if you imagine in a non-pregnant state, you're looking at Christmas lights that are flashing randomly. That's the non-pregnant state. When you go to pregnancy, they all flash together. Everything pulses together. And it's that dramatically increases vasodilation. What then happens is in mild preeclampsia, where you start to see some inflammatory cytokines, is that some of those gap junctions get phosphorylated and closed. You lose that connectivity. And if you go to severe preeclampsia, where you've got failure to vasodilate, hypertension, and edema, you've not only closed them, you've got to the point where the cells are pulling apart from each other. They can't even hold on to each other. This is why you've got the edema. Okay. So we're going to test the hypothesis that the Th17 and Th1 inflammatory cytokines can actually mediate this process. And more importantly, there is an emerging field that says that if you expose these cells long-term to cytokines, they will start to become non-professional antigen-presenting cells. These cells actually cease to be what you consider endothelium. They start to take on another role, and they express antigen-presenting proteins on their surface, the so-called CD molecules. Okay, CD40, CD8, etc. And these allow these endothelial cells to bind and interact with immune cells. And that raises the local concentration of these hormones. So we're going to be looking for that. We have done transcript analysis, looking at different treatments. And I'm sorry about the title bar, but the transcripts that have come up in this yellow circle here is the TNF treatment circle. If we treat uterine artery endothelial cells with TNF, we see a whole bunch of transcripts change. Many of those, the take-home message is that many of those are antigen-presenting innate immune cell proteins, even though these are endothelium. They're 100% pure endothelium. Not a matter of contamination. They're taking on that property separately. The others are endocrine. There are hormones such as cytokines, chemokines like CCL5, CXCL10, growth factors, cytokines, interleukins. And some of them are extracellular space, like metalloproteases that can digest the extracellular matrix and cause the cells to come apart and for edema to begin. TNF is a stimulator of all these changes. TNF does significantly and selectively regulate the secretion from endothelium of interleukin 6, 8, and CXCL10, as well as interleukin 7 and interleukin 12. The first ones, interleukin 6, 8, and CXCL10, are all markers of preeclampsia that have been reported recently. IL-7 and IL-12 are known to stimulate the immune system to move towards Th17 lineage. And now we start to see the interaction already. So the combined data is consistent with studies in humans and is actually being paralleled by work by Derek Belt in Project 4. And it is consistent with the observed changes in the circulation in preeclamptic subjects of TNF, interleukin 1b, interleukin 6, interleukin 8, and the chemokines CCL5, CXCL9, CXCL10, and CXCL11. So of the additional antigen-presenting transcripts that have actually been presented by other people, and reviews have been written, and this is a figure from one of them, many of the surface antigens like CD40, CD155, et cetera, many of them have been detected already in our cells, and some of them are increasing in response to TNF. So we are seeing this phenomenon in the uterine artery, that cytokines can drive them to an antigen-presenting state. So the question is, what further degrees of antigen presentation can be promoted? How do the state changes compare to freshly isolated HUVEC? Because it does look like the changes in the umbilical cord parallel the changes in the mother to everything we've seen so far. And what secretory and antigen presentation properties do they have that would give us a clue as to which T cell population they would bind? Because they will be specific. This is very recent data. We've only done it once, and it's taken us 6 months to develop this multi-panel FACS method for looking at surface antigenic proteins. Every one of these is a fluorescent antibody binding to a single cell at the same time. We can measure them simultaneously. And you can see that there are a large number of CD proteins that we're screening for. Few of them are negative, most of them are positive. And more to the point, the red and the green means that they either went up or down relative to control when we treated with different agonists. And the most important one we're looking at here is TNF, this one here. So our aims are, the first is to define the state and fate and functional changes in ovine endothelial cell model. We're going to use our sheep cells to begin with to artificially stimulate them and look at what changes happen in response to known agonists and relate that to transcription factors and signaling kinases. And our second aim is to look at how those changes we've invoked artificially in the uterine artery endothelial cells are paralleled by what we actually see in isolated HUVEC from normal and preeclamptic pregnancies. How much do they have in common? We're either going to come out with an agreement, in which case we're perfectly right, or we're going to come out with a disagreement, which means we've missed it somehow and we've got to do more work. So, Project 2, Alex Stanek-Kostek, he takes this much further. He goes into the decidua, looks at the immune cell population, and his sort of central premise is that there's pro-inflammatory plasticity of innate and adaptive decidual lymphocytes that are directly interacting with maternal endothelium, and that this is the cardinal feature of preeclamptic pregnancy. So we're going to use a method and an analytical technique, both of which are cutting edge. He's actually going to map in decidua all of the cells and do single cell seq, RNA seq, and site seq on either the tissue or on the isolated immune cells and compare the results. You've got to bear in mind that the endothelium data will also be coming from Project One. So he will have reference data for pure immune cell populations and reference data for endothelial populations, so that when he looks at a heterogeneous tissue, he can deconvolute the data to work out for each punch that he makes into the tissue, how much is endothelium, how much is immune cells, and which particular cells are they? What state are they in? Okay. So this is an overview. Obviously, we have the blood in the crypt, and then we have the fetal side of the placenta here, maternal side here. The stroma contains maternal immune stromal cells, fetal placental trophoblast, and then there's altered immune maternal vasculature. Within this space, there will be immune adaptive lymphocytes, T and B cells, macrophages, dendritic cells, endothelium, there's trophoblasts. Okay, so the idea is to ask which immune cells interact with the endothelium. This is restating what I just said. Is the preeclampsia associated with altered transcriptional programming of the immune cells? Do the immune cells exhibit stable epigenomic changes? What is the spatial context of interacting immune cells and endothelium? Do immune cells committed to inflammatory programming traffic between the decidua and the systemic vasculature? The systemic data will come from Project 3, which I'll get to in a minute. And do directly, this is the key, is do you see evidence for change, direct interaction in preeclamptic subjects between the endothelium and attached immune cells? Do they drive each other crazy? Like the marriage that never ends, but everybody's always shouting at each other. It's that. Okay, this is the odd couple, right? So Project 2, this now gives you the details. I will just say with you, anyone who knows Alex, this is classic Alex. I won't go through it. Love him to death, but Alex, come on, please. The truth is, this is what Alex, and all joking aside, this is what Alex and Irene have to do to process the data and deconvolute it. And it can only be done in the context of this program project grant, because it's only in this context that we have the data on pure cell populations to compare, run on the same machines with the same methodology. So he will be able to do this. Afterwards, everyone will say, well, it's easy, anybody could have done that, but we haven't got there yet, right? The reality is, this is a section on the left. This is a picture of a tissue section of the maternal fetal interface, right? And what you do is you isolate the cells individually and you sequence them, and you get the surface markers from site-seq with antibody markers. But then, rather than saying, I need an endothelial cell, let's go find one, you don't do that. You grid the entire tissue sample and punch every circle and sequence every circle, and then deconvolute the data. And basically, you say if two cells were in the circle, then they were interacting. Okay, so it's a lot of data that he's going to have to get. What data do we have that this is going to work? Well, obviously, that technique is horrendously expensive. You know, you do one experiment, it's $70,000. So that's a big deal. But what we can do is we can look at the isolated PBMCs, peripheral blood mononuclear cells, from decidua and from systemic. And you can sort them and look at the surface markers by FACS to see if at least what we're talking about is reasonable. And he has done that and created matched cell data. And and looked at gene expression with 11 surface antibodies that would be usable in this more sophisticated technique down the road. And basically what he shows is that data for integrated CD4 expression, CD8A expression, and PDC CD1 expression. When I say expression, I mean the detection of transcripts in those cells, the RNA in those cells, matches the distribution of surface proteins reliably. Not only that, but the antibodies give more clear, concrete data than the transcripts do. Okay, so far, even whether it's, consider this negative and this positive, you've got black and white results here. Here, you have a mixed distribution, and sure enough, with the surface protein, you have a mixed distribution. Each point on here is a single cell. Okay, so we're seeing consistency in the idea of comparing transcripts and surface proteins. And then over here, what we see is that the distribution, I spoke to Alex about this yesterday, I hope I get this right. The distribution of commonly presented markers of immune tolerance in the immune cells from 3 patients are all distributed similarly, even though they're different gene products in the T cell population, or in the PBMC population to be accurate. So these so-called TCR CD3 clonotypes are similarly distributed, and he's detecting them reliably. So the tools work and can be applied for us to characterize all these cells, either from pure populations or from tissue extracts. So the specific aims for this are to determine pro-inflammatory transcript signatures of tissue innate and adaptive lymphocytes in healthy hypertensive and preeclamptic pregnancies. And specific aim 2 is to determine spatial architecture and verify the predicted lymphocyte endothelial interactions that get to be observed in vitro. And when you bear in mind that data from other projects will also tell you whether surface proteins are on the endothelium that will allow the attachment of that particular T cell population, we can be pretty certain that that is a real interaction or it isn't. Okay. So project 3, this is where we move to the systemic vasculature and look at the immune cell population. We can monitor the fate changes in circulating immune cells in response to healthy and hypertensive pregnancies. And this one is a longitudinal study. Everything else is at the time of delivery, but this is where we get longitudinal. So this is the one where we look at, or they look at, longitudinal blood samples from the first clinic visit through to delivery. Looking at the circulating immune cells of pregnant women with preeclampsia, hypertension, to exhibit altered transcripts and function that reflects the altered inflammatory state, or to put it another way, the T cell population and activation state of that population, and decidual immunome, and is predictive of compromised systemic and uteroplacental blood flow. The point to understand here is that we believe that the decidual cells are getting out into the immune cell population, and therefore we would be able to do a blood test on a pregnant woman to look at her risk, because we think there's exchange of these cells all the time. And we now need to either prove it or shut up and go away, right? Fate of maternal immune cells is influenced by these inflammatory cytokines, growth factors, and interactions with the endothelial cells. And a subset of these changes are reflected in the immune cells that traffic in the circulation. So we're going to look again using the same technique, surface protein markers and transcriptome analysis, the changes in transcriptome and phenome of circulating immune cells as potential biomarkers early in pregnancy, 12, 14, 20 weeks, to tell you what's going to happen in the outcomes phase before it happens. This is some preliminary data. This is a list of transcripts, a heat map of transcripts that have been detected in PBMCs from women who are either control, which is this first 3 columns here, or preeclamptic, which is the second 3 columns. And I would point out that some of the markers that are being listed as altered in these cells are also chemokines and surface markers. And there are, in fact, 300 other genes that need further validation that have been preliminarily identified. And in a separate set of samples, differential gene expression has also been observed in pregnant women at higher risk of hypertensive pregnancy. So the other thing is that Dinesh Shah just concluded a year one, and we already have collected these cells, these peripheral blood mononuclear cells, from that population of women. So we have a set of women now where we have these PBMCs from primarily normal pregnancy, along with very detailed and sophisticated measures of blood flow by MRI as well as by ultrasound, and complete endocrine panels on all of those for cytokines and chemokines and growth factors. So when you take that very strong control set, it will be easier to find a deviation from normal in the preeclamptic subjects. The goal of this project, therefore, is to collect samples from the first clinic visit of a high-risk patient through to the end and determine if the hypertensive pregnant women exhibit longitudinal changes in transcriptomes and surface markers of their circulating immune cells, determine if the changes in the transcriptome and the surface markers reflect the decidual data from Project 2 from Alex. Do they agree or disagree? Are we seeing the same cells or a different set of cells? That'll help us understand whether there's really free exchange between decidua and systemic or not. Or do they just go into the decidua and stay there for 9 months? And then the final point will be to look if the data from those parts of the study relate to something that Dinesh is doing with Jenna Racine, which is to look at whether the forearm blood flow changes that reveal the extent of dysfunction of the vasculature in the mother correlate to the perturbations in the immune cell population and the clinical presentation of preeclampsia. Okay? Because if that's true, forearm blood flow could become a standard clinical technique. Then the final one is we move into the umbilical cord. This one is a project led by Jing Zhang and Derek Belt. And this is looking at endothelial cells, looking at HUVEC, some of which I've described. But the other part of this is it's looking at actually modeling direct interaction between immune cells and endothelium. And what we're looking at is whether pro-inflammatory immune cells can actually drive endothelial dysfunction in the same way as if we add exogenous hormones in vitro. And whether the abnormally derived endothelial cells from, say, preeclamptic subjects can cause immune cell fate changes in preeclampsia. And the third part is a pilot project. There are some similarities in the literature between the signaling and the impact of aromatic hydrocarbon receptor agonists, which are, for want of a better word, are things like DDT variants, the toxic compound DDT, the pesticide. Exposure to DDT and its variants does actually cause endothelial damage. And much of the damage that is caused is similar to the damage we see in preeclampsia. So there's some pilot work going on in this project looking at signaling related to aromatic hydrocarbon receptors. The data says, if you look at the top left A here, the data says that if you look at, I described earlier on how the endothelial cells can coordinate the calcium function. They either look like Christmas lights randomly flashing or they coordinate together. This is the percentage of cells at a coordinate function. And what happens if you introduce into that in vitro system simply exposure to immune cells or immune cell lines. And all of these immune cell exposures to different degrees, these are freshly drawn cells, these are frozen and then recovered cells. All of them reduce calcium signaling just by having them in the media for a few hours. And then the second thing is that this is as potent an inhibition as TNF. Okay, so yes, exposure to immune cells is as potent as we have been modeling with hormones. And probably for the same reason, the immune cells are secreting these hormones directly onto the cells. Furthermore, if even if you get cells from a patient that don't give much of an inhibition, if you then activate those immune cells first and do the same experiment again, you get an even worse inhibition. And here you can show that if you simply increase the number of immune cells, you get increasing inhibition of the endothelial cell function. So it makes sense that if you were to attach more cells, you would see more damage. Okay, it is literally cell number dependent. Derek has actually worked with bioengineering to create a new model to look at interaction of cells. What he does is he makes a gel, and it has a channel in the gel that is like a pipe, and you can put liquid through there. And then you introduce cells and you grow them until they coat that pipe, and what you get is an endothelium-lined pipe, just like an endothelium-lined vessel. When those cells are stable after several days, you can then introduce immune cells, let them sit for a few hours, and then wash the unattached cells away. And this allows you to see the attachment of immune cells to endothelium. The ones that attached remain, the ones that didn't are washed out. You can see on this figure here, there is always a certain amount of attachment that goes on. Immune cells will always attach to a certain degree to endothelial cells. But if you pre-expose the cells, the endothelium, to TNF first, and then do the experiment again, you see this dramatic increase in attachment. It becomes continuous and solid. And this is a quantification of that change. The other thing is if you put the cells in and you put in, instead of the PBMCs, you put in the cytokines, you can show that if you put a red dye, which is shown here, if you put a red dye into the channel and then you do the experiment, let it sit, where the endothelium is intact and you add nothing into the media, the red dye remains confined to that tube. But if you put the hormones in for cytokines, you see that that dye spreads because you're eroding the endothelium connectivity and you're getting the edema-like effect. And you're seeing the same thing as you see in edema. This red dye is now spreading to places where it shouldn't be. It's getting beyond the endothelium. You can also reproduce this with introduction of immune cells and reproduce the same result. And the final thing is, we talked about this, what we call Christmas lights versus, pulsing of all the calcium. This is actually a graphical representation of that. 100% is the number of cells, all of them flashing together, every cell coordinate with every other cell at the same time. It's the equivalent of crowd noise at a sports event versus they then do a chant together. So you've got a lot of noise, but it's all random versus, everyone goes to wave and go, whoa, and you hear the sound coordinated with all these people together. It's much louder than the crowd noise where everybody's talking, okay? So it's like that. So 100% is like everyone's singing together, and 0% is they're all just sitting there talking about what did you do at the weekend, right? So you hit the cells with an agonist to get the calcium response going, and there's always 100% because the agonist hits the cells. But the question is, long term, do they hold that calcium response together or not? And in normal cells from normal cords of healthy pregnancies, you see 70% of the cells are singing together. But if you expose them to cytokines, that goes down to 10%, 10 to 20%. They can't respond after the exposure to cytokines. You put PBMCs in there and allow them to secrete their hormones and attach to make those hormones highly effective, and you get this impaired calcium synchronization response. You activate the PBMCs and it's even more potent. Now they're secreting more hormones and there's more damage. But this is the interesting one. If you look at the preeclamptic cells, you don't do anything other than take preeclamptic cells from a preeclamptic pregnancy, try to do the same experiment, they don't coordinate at all. They're already so damaged, they cannot coordinate. This final project is the pilot for aromatic hydrocarbon receptors. And I'm not going to go through this in detail because it's I think, too complicated for me to do it in the time we have. But I will say that there are clear effects on HUVEC of normal pregnancy, which aromatic hydrocarbon receptor ligands can cause to change in the same way as we would see with cytokines, and the same way we would see with preeclampsia. And ITE, just anyone who wants to look at this, ITE is a ligand for the aromatic hydrocarbon receptor. So, and you can see that there are studies where, I believe this is animal studies, where fetal weight is reduced by ITE and placental weight is reduced by ITE exposure if you feed this to the pregnant animal throughout gestation. So Project 4 aims then are to determine how the activated overactive immune response drive endothelial dysfunction and differentiation, change in antigen presenting state in hypertension in coordination with Project 1. And the second is to see if you alter the endothelium and then you put in normal PBMCs, do they become abnormal? Is endothelium actually a trigger, or is it the victim? Is it the perpetrator or the victim? Okay, tune in on 7 o'clock on CBS and you'll find out. And then aim 3 is a pilot to look at these aromatic hydrocarbon receptor ligands and whether they actually could play a role in the preeclamptic process. This was for NHLBI. For you, it's a very boring slide. You have to have 3 cores in a program project. We will have an administrative core. We will work with the research office, with Andrea, to control the budget and so on. We will also have a human subjects core, which is core B, which I'll get to in a minute, and we'll be running administration with the divisional secretary, Cindy Goss, as well. So between Andrea and Cindy Goss, we'll get everything done. Core B is more relevant here. It's the human subjects core. This is going to take quite a bit of work. Derek Belt is actually the PI of this core, working closely with Dinesh Shah, who's an MFM, Manish Patankar, a coordinator. We were, at the time we put this proposal in, that was Rosalina Landeros, but now she has left and we would be working with the new replacements. And we would appoint a clinical research coordinator from the grant. The sample processing, again, time has moved on. That was Jason Austin and Beth Bird, but now is Serena and her new staff that are coming online this week. The number of subjects, I haven't covered that yet, and I thought I'd cover it here. Over 5 years, we're going to be looking at recruiting 200 subjects. We would break that into 2 groups. The first group is very easy because you already have the diagnosis, you already know what happened, and you're only analyzing at the end point to see what the outcome was. So 100 subjects are simply controls, early preeclamptics, late preeclamptics. We need a minimum of 25 in each group. So we're going to recruit 100 to get 75. The other group is more difficult, and that's where you take a prospective group, people who are at high risk, and you start sampling from their first clinic visit to the end. And we're going to collect 100 of those, and then we get what we get, early and late preeclamptic. The chances are we're going to have more early preeclamptic than late preeclamptic, but that is fine, because we can either look at the late preeclamptic to see if they agree with the other projects. We can look at the early preeclamptic to see if they disagree. Is early preeclampsia really a different condition, or is it the same condition with an initial trigger that is different? We'll be able to actually give you information on that by the time we're done. And then core C is pretty cool. We're almost done here. Core C is the analytical core, using the artificial intelligence methods, analyzing the data, kind of data we've shown you, and other data. There are 2 advantages to this. One is Irene is blisteringly intelligent, has fantastic staff, and I won't even pretend to know what she knows. I couldn't do it even if I tried, so I'm not going to pretend. The other thing is you have an independent group of people analyzing your data, and they can analyze it blind. So that has some advantages from rigor and reproducibility standpoint. And the third point, which I must say, because Irene will kill me if I don't, is develop new methods. This is right at the forefront of research right now. Single cell seq and single cell site seq is right at the forefront. This may be the first time it's ever been applied to preeclampsia, certainly in this context. There are going to be multiple omics data sets, and she can not only analyze them individually, she can analyze them together. Okay. So some examples, bulk RNA-seq, which is simply what most people have done for 10 years, CyberSort, single-cell RNA-seq, and then the variant where you add the antibodies to surface markers, which is site-seq. And then, of course, we can look at T-cell receptor sequencing, spatial transcriptomics, which is what Alex is doing. And of course, for all of the projects, we do a certain amount of fax analysis. We have to analyze that data. The methods development, well, clearly, you know, this is what Alex has for breakfast. This is what Irene has for lunch, right? This kind of stuff, deconvolution. Theoretically, in principle, it's very easy to understand. In practice, it's incredibly hard to do, and she's going to do it. I have no doubt she will. And then, of course, there are more advanced machine learning methods to look across omics and combine it with data sets of clinical data. Actually put into that algorithm the clinical outcomes and ask which theoretical data most predicts clinical outcome and which has the greatest role in predicting the clinical outcome. Is it the clinical data? Is it your dipstick protein? Or is it the expression of CD40? Okay. And we won't know until we finish. $10 to the winner, right? Okay, thank you. That's basically it. And if you have any questions, I'd be happy to take them.
All right, anyone, any questions here in person, or feel free online to go ahead.
Good morning, and thank you for that presentation. I was just wondering, since you're building this cohort of women, whether there's a plan to look at immune profile and endothelial function in folks who end up having persistent hypertension after a preeclampsia diagnosis versus those who basically resolve their hypertensive issues after pregnancy.
The biggest driving factor in what we can and cannot do, it's probably no surprise to anyone when I say this, is money. Okay, so I would love to have a trial with 5 centers across the country and recruit 1,000 control patients, 1,000 preeclamptic patients, 500 early, 500 late. I'd even like to just collect, hypertensive patients, hypertensive diabetic patients, and diabetic patients and compare. And if I did that, it would probably cost us, I'm not joking when I say $50 million. Okay. The PPG budget depends on how you look at it, it's between 11 and $13 million. So that's the biggest limitation. Having said that, your question is not only right on the money, I've talked to Cara about that, Cara Hoppe, because that's what she does and does it very well. And we have talked about actually whether if we were to do this, we could also, during the PO1, submit a grant to do the immediate follow-up on the patients from this study as well. So, it would take time to get it funded. You wouldn't be able to do it in years 1 and 2, but a PO1 is always 5 years. We might be able to do it in years 3, 4, and 5. And it'd be very interesting to see, whether the, well, obviously, 10 weeks after pregnancy, there's no placenta, right? But you got the placenta data from when the baby was delivered, so that might be predictive. And of course, we can continue to blood sample, so we can look at the systemic immune cells. We can continue to do brachial forearm flow. So yeah, it would dovetail really nicely into this study if we could get it funded. Stunned you all into silence, right? Yes. All right. Okay. Do you want to ask anyone online or not?
We're going to just open up the chat here and take a look. Feel free if there's anyone online. We'll give you a moment to unmute here.
Did you get the code back, Bagler? Well, this is basically the difference between systems biology and, traditional do a Western blot on a cell line and tell everybody what happened. I mean, this is really getting into the patient world, into the in vivo situation. Everything we're doing in vitro is to tell us the validity of the data in vivo. That's where it's different.
And that's where the advances are thankfully going forward, where we can do these sort of studies, which just 10 years ago, we couldn't have thought of doing.
Yeah.
So thank you for that wonderful presentation. I was going to ask about the numbers and how did you come up with the 200? I mean, I can't imagine doing a power analysis here. So
it's a trade-off. I mean, it has to be a trade-off.
Right.
Because there are only so many hours in a day and so many patients you can recruit. We're deliberately keeping the BMI below 40 because we don't want complications from extreme obesity. We're going to have a upper cutoff on age. We're trying to look at the main population of the patients that present at America. And yes, that's limiting. But like I said, no one's ever done this before. This is going to be the reference data set.
Right.
This is the reference data set by which you can then go on and say, does this particular population, does this have even more extreme symptoms? Or if they have more extreme symptoms, are they related to antigen presentation or not? And that could be anything from studying a socioeconomic group difference, ethnicity difference, age difference. Comorbidities like obesity, diabetes. You can do them. Once you've got this, you can do everything else.
Excited. Thank you for sharing that.
Yeah. Yeah. Thanks.
We'll wrap it up here.
Yeah.
Thank you very much.